This is the pre-lab lecture video for Iron Analysis Lab for Chem 1B for Fresno State. Now, as we start this lab, I'd like to start off with discussing how we see color and why things appear colored. And they appear colored because they reflect the colors that are not being absorbed. Thus, things appear to be the complementary color that they are strongly absorbing. So let's say we have a compound that is strongly absorbing yellow light. So if it's strongly absorbing yellow light, all the other colors are being reflected back. So we see mainly the purple light that's being left over. So if something's absorbing yellow, we are seeing the complementary color, so it would appear purple to us. Now in the lab, we're going to be making an iron phenanthylene complex. And that iron complex absorbs strongly at 508 nanometers. So that 508 nanometers is right here in the green-blue region of the electromagnetic visible spectrum. So if it's absorbing strongly in the green-blue region, so let's say right around here, we would expect our compound to appear red-orange, and that's what we'll actually see. So our compound will absorb at 508 nanometers, which is a blue-green wavelength of light, so we will see a red-orange compound that's being formed. Now when we see colors, there is an electron in a ground state orbital. And if we give that compound energy, that electron can be excited. Now when we look at the difference between the ground state and the excited state, that difference here that we're representing in this height, that's an energy difference. And that energy difference can be related to this equation here where we have hc over lambda, where h is Planck's constant, c is the speed of light, and lambda is the wavelength of light. So if you have a wavelength of light that matches, that gives the right energy, that electron that's in the ground state will be excited into the excited state. So it will absorb that wavelength. So what we see in our compound that absorbs at 508 nanometers, once we give it that wavelength of light, that's enough energy to cause that electron to be excited from the ground state to the excited state. Now once the electron's in the excited state, it wants to fall back down to the ground state, and the energy is released in thermal vibrations as the electron falls back down to its ground state. So the key thing here that we want you to take away is that colors are caused by electrons being excited, and that excited electron has to be excited by energy, and there's a wavelength of light associated with that energy of light that's being absorbed. And the color that we see is the complementary color of the light that's being absorbed, because we see the light that's being reflected back. So we're interested in the colors of our compound because the intensity of the color, or how much light that the compound absorbs, will be related to the concentration. So we're going to use Beer's law. And Beer's law states that the intensity of color or absorbance of a substance is proportional to the concentration of a substance. And so this is our representation of Beer's law, where A is the absorbance and E is the molar absorptivity. And this is a constant that depends upon the nature of the colored species. B is the path length of light traveling through the cuvette. And C is the concentration of the colored species. So E is just a constant related to the particular compound. B is the path length of light that goes through it. So the smaller the sample size, the less light that can be absorbed by the sample. So this is why when we look at the air that we breathe around us, we really don't see anything being absorbed, even though we know that there's pollution. But yet when we look in far off distances, we try to see the mountain range from us here at Fresno State. Sometimes the mountain range is obscured by pollution. That's because there's a longer path length for that light to pass through, and so those pollution molecules are absorbing more of the light, so we see that path length dependence there. Now also too, what we're going to be focused on this experiment, because we're going to keep the molar absorptivity is going to be a constant, and the path length is going to be kept the same. So these are going to be kept constant, but we are going to vary the concentration. So increasing the concentration causes an increase in absorbance. So you'll be producing graphs similar to this here. So here we have the iron calibration curve. On the x-axis, we have the concentration of the iron in molarity. And on the y-axis, we have the absorbance at 508 nanometers. Now, one of the places you may have seen this example of concentration versus absorbance in your everyday life, think back to when you were a kid and you made Kool-Aid or your mom made Kool-Aid for you. The more flavorful the Kool-Aid was, the darker it was in color. I don't know if you went over to some of your friends' house and their mom made the Kool-Aid weaker, they added more water to it, is more dilute, so it wasn't as intensely colored. So maybe your fruit punch Kool-Aid was light pink and not as flavorful because there wasn't as much Kool-Aid powder in there. Or maybe it was really dark red and it was really flavorful because there's a lot of Kool-Aid powder in there. So the higher the concentration in there, the more intensely colored it is, which means the more light that it's being absorbed. So the objective in this lab is that we're going to take some iron supplement pills, basically some vitamins, and an unknown solution given to us by the stockroom, and we're going to analyze the concentration of iron in it as iron 2 plus using colorimetric analysis. Basically, the more intensely colored it is, the more concentrated it is, and we're going to be able to quantitatively measure the concentration of the iron. Now the iron in our sample is either in the iron unknown sample or in the iron pills. That iron could be in two different oxidation states. It could be in the iron 3 plus oxidation state, or it could be in the iron 2 plus oxidation state. But for this experiment to work properly, we need it all to be in the iron 2 plus oxidation state. So to do this, we are adding hydroxylamine hydrochloride. So this is why you're adding the hydroxylamine hydrochloride in there. You're converting any iron 3 plus that might be in there, and you're reducing it to iron 2 plus. Now the iron in there is going to be colorless, but we're going to add this orthophenanthroline to the compound, to the iron. And what's really cool is that once the phenanthroline binds to the iron and makes this iron phenanthroline complex, this iron phenanthroline complex is intensely colored. It has a really pretty orange-red color to it. So we start off with this colorless compound, and when the iron adds to it, it makes a compound that is orange-red in color. Now this orthophenanthroline is a bidentate ligand, so those lone pairs can find an iron and bind to it. And the iron can accept 3 of these phenanthroline ligands. And this makes an iron complex. And we're going to learn more about complexes and transition metals when we get into our transition metal chapter later on in the semester. But this is kind of a bit of a preview on our first iron complex. So again, we're adding the phenanthroline to make the iron colored so that we can easily measure the concentration from the intensity of the color. So one of the things that we'll need to do is we'll take several solutions of known concentration of iron, and we're going to measure the absorbance of them, and we're going to create this trend line here. So this is our calibration curve, and from the calibration curve we will get a trend line. So this y equals mx plus b, and from this we'll be able to calculate the molarity of our unknown solution. Because remember, let's go back to our Beer's law where A equals EBC, and the molar absorptivity and the pathing are going to be kept constant. So we can think of the absorbance as the y value, so our dependent variable, and the x as being the independent variable, and that's going to be our concentration. Now when we plot our curves, we do not want to force the line through the intercept. We want to have it in the form of y equals mx plus b. So this b here is going to be related to some of the error in the lab. But the better the lab is performed, the closer this y-intercept is going to be to 0. So if the concentration is 0, we'd expect the absorbance to be 0. Now I just pulled this picture off the web, and we have different solutions of these iron phenanthroline complexes. So I would expect that this solution here, which is lighter in color, would have the lowest concentration, and here we get a little bit darker, and this last flask is darkest in color, would have the highest concentration. And remember, the more dilute one is going to be less intense in color, so this would have the lowest concentration. The trend line made with the known concentration allows us to calculate the unknown concentrations from how much light they absorb at 508 nanometers. So once we get this trend line and we find out where, let's say, our unknown absorbs at 0.13, we'll be able to calculate the concentration based on that trend line. So if we know the Y value, we can solve for X. So this is just to give you an overview of what we're going to be doing in lab. So part A, we're going to be preparing the commercial iron supplement solution. So one group in the class is going to be doing this first part. Basically, you're going to be dissolving the iron pill in 2 liters of water and adjusting the pH of the solution. Now, while one group is doing this, the other students in the class should be starting on part B. That way nobody's sitting around twiddling their thumbs. So in part B, we're preparing the colorimetry standards. So those are those standard solutions that we know the exact concentration of the iron. And when we know the exact concentration of the iron, we can measure the absorbance of those solutions. So when we're preparing those standards, there should be 3 burets that are prepped. There's going to be the iron 2 plus standard, the 0.25% of the phenanthrene, because remember the phenanthrene is going to bind to the iron to make the nice orange color. And then the last buret should be the 10% hydroxylamine hydrochloride. Because remember that hydroxylamine hydrochloride is there to convert any iron 3 plus to iron 2 plus. So once the 3 burets are prepped, then you prepare the 6 solutions of different concentrations from the 40 parts per million iron standard solution that's given by the stockroom. And so the directions on how to prepare those solutions are in your lab manual. Now when you prepare those solutions, you want to use the same 50 ml volumetric flask. And the reason being is that there are not enough flasks for everybody to have 6 different flasks. So make your solution in the 50 ml flask, pour it into a beaker, and then clean it out and use that flask over again. So once your colorimetry standards are prepared and some group has prepared the 2-liter solution, we need to go back to part A and prepare a 50 ml sample of that solution in a volumetric flask. And so in that 50 ml volumetric flask that contains the sample, there's going to be a 1.5 ml aliquot from the 2-liter iron pill stock solution. There's going to be enough sodium acetate to bring the pH to about 3.5. And then we have to add the hydroxylamine and the orthophenanthrene. So once we have all of our solutions prepared, we're going to prepare our final dilutions for our blank, the iron pill, and the unknown iron solution. Then once all the solutions are prepared, we use the colorimeter to measure the transmittance of the light at 508 nanometers through all the samples. And then after you do that, then it's time to do calculations in all your work in Excel. So we have provided an Excel template for you on Blackboard. So that way everybody's looks the same. All the labels are given for you. You just need to add in the values that you used and calculate the necessary values. Now the yellow portion in here, the only reason why this is yellow, this is for the grader. The yellow lets the grader know that those are the values that he has to put into the grading sheet, and all the other numbers are generated for the grader. So that's why you have the yellow values. But all the other blank values you'll have to fill in. So make sure you put in your name and your lab partner's name, how many drops of the sodium acetate solution that you added to each of the sample. You'll measure the percent transmittance, and then from the percent transmittance you'll calculate the absorbance. You'll also calculate the concentration of the iron 2 plus in parts per million and the concentration of the iron 2 plus in molarity. And from this data here, when you plot the absorbance and the concentration, you'll calculate the slope and you'll calculate the y-intercept value using the functions in Excel to get your trend line. And then from your trend line and the absorbance of your iron stock solution and your unknown iron solution, you can calculate the concentrations of those. And so you'll put the remaining calculations in here. and then make sure you add your graph in this section down here where it says Insert Graph here. Now when you put in your numbers in the Excel, don't just calculate on a scratch paper and type in those values. What we want you to do is to use Excel to calculate those values for you. And there are two main reasons why we want you to do this. First of all, it minimizes rounding errors because it just keeps the numbers when you refer to the different cells. Secondly, if you realize you've made an error at the very beginning, at the end, you can go back and change the cell and it will update all the other cells after that. So if you make an error, it's easier to correct than if you had done all the calculations by hand. So make sure you do the calculations using the cell functions in Excel. Now part of this lab will require quantitative transfer, and quantitative transfer simply means that all of the material to be transferred from one place to another must make the trip. For example, every particle of solid must be transferred from the weighing paper to the clean beaker. If you are transferring a solution or heterogeneous mixture to another vessel, rinse the container with solvent to be sure that the transfer is quantitative. The rinsing should be transferred to the second vessel along with the rest of the mixture or solution. Now here we have a picture of a burette, and hopefully you've used a burette before, especially in your Chem 1A. And a burette is used to deliver a solution in precisely measured variable volumes. And burettes are used primarily for titration to deliver one reactant until the precise endpoint of the reaction is reached. Here we're going to be measuring out precise volumes. We're not going to be doing titrations, but we will be using burettes. Now to fill a burette, close the stopcock at the bottom and use a funnel. You may need to lift up on the funnel slightly to allow the solution to flow in freely because it may create a bit of a suction there and you need to let some of the air out. You can also fill a burette using a disposable transfer pipette. This works better than a funnel for the small 10 ml burettes. Be sure the transfer pipette is dry or conditioned with a titrant so the concentration of solution will not be changed. Before titrating, condition the burette with titrant solution and check that the burette is flowing freely. Make sure you don't have anything lodged in it or causing anything to block the flow of your burette. Now to condition a piece of glassware, you want to rinse it so that all surfaces are coated with a solution and then drain it. Conditioning 2 or 3 times will ensure that the concentration of the titrant is not changed by a stray drop of water or whatever may be left behind in the burette. Also, when you're preparing your burette, check the tip of the burette for air bubbles. To remove an air bubble, whack the side of a burette tip while the solution is flowing. And whack may be a strong term. Simply tap it, and you can progressively tap a little bit harder to help dislodge the air bubble. If an air bubble is present during the titration, volume readings may be in error. The volume of that air bubble may change, or the air bubble may become dislodged during the titration. So make sure you get rid of it before you start. Also, rinse the tip of the burette with water from a wash bottle and dry it carefully. After a minute, check for solution on the tip to see if your burette is leaking. The tip should be clean and dry before you take an initial volume reading. Now, reading the burette is very important. When your burette is conditioned and filled with no air bubbles or leaks, take an initial volume reading. A burette reading card with a black rectangle can help you make a more accurate reading. So this makes it easier to see that meniscus, because first of all, you're not having to worry about seeing things that are behind the meniscus. You have that white background there, but also the black square reflects back up so you can see that bottom of the meniscus much easier. So make sure you read at the bottom of the meniscus and be sure your eye is at the level of the meniscus. You want to read it at eye level, not above or below. Because if you read from an angle rather than straight on, this is going to result in parallax error. Also to remember that burettes are recorded to the 0.1 mil place, so you need to read it to, you need to estimate the next decimal place. So the volume is always read to 2 decimal places on a burette. And you want to deliver the solution to the titration flask by turning the stopcock. The solution should be delivered quickly until the desired volume is reached. Now let's discuss how to use the Spec 20. When we're setting up the spectrometers, make sure that the instrument is turned on. Here we use the left hand knob and we turn it to the right so that it turns on, and you want to allow 10 minutes for warming up. And at the top here, we see a top view of it. Dial this into the wavelength that you want to use. So we're going to be setting this to 508 nanometers. Then you need to calibrate the spectrometer. The sample compartment here must be empty and closed. Turn the left hand knob to obtain a reading of 0% transmittance. So up here, make sure that it's at 0% transmittance. And we want to use the mirror behind the needle to avoid parallax error. So think about when you're driving and you're watching the speedometer of the person next to you. You may read something differently because you're seeing it at a side versus the person that's looking directly on. So this is why we want to use this mirror to help avoid that parallax error. Now wipe the cuvette with a dry Chem wipe to remove drops of solution or fingerprints. And we want to use a Chem wipe rather than paper towels so that we don't scratch the cuvettes. Now in lab we're actually going to be using test tubes rather than cuvettes. Cuvettes are specially made for the spectrometers. They're very precisely made, but they're also very expensive. So we are just going to be using test tubes in place of cuvettes. Now when you insert the cuvette, you need to always input it in the same way. So you might want to make a little line at the top of your cuvette or your test tube so that that line is always positioned at the front of the instrument. So always put it in the same direction. That way, in case the thickness of your glass may be thicker on one side versus another, it's always going in the same way. So you insert a cuvette filled with solvent in the sample compartment, and this is called the blank. Make sure again to line up the mark on the cuvette with the line on the sample compartment. Then you close the cover. Then turn the right hand knob to obtain a reading of 100% transmittance. So the left is for 0 without the sample in there, and the right is with the blank in there at 100% transmittance. And you may need to repeat this process a couple of times to make sure that you consistently get the same readings. Now also, if you change the wavelength or you change the solvent, you have to recalibrate the spectrometer. But in this experiment, once you calibrate it, you shouldn't have to recalibrate it again because we're not going to be changing the solvent and we're not going to be changing the wavelength. Now finally, to analyze your sample, insert the sample cuvette and read the transmittance value on the scale and use the mirror behind the needle to avoid the parallax error. Rather than reading the absorbance value, we want to calculate the absorbance from the percent transmittance. So you will record the percent transmittance, and then in Excel you will calculate the absorbance from the percent transmittance. And this minimizes the error associated with reading values on a logarithmic scales. So the equation for this is absorbance equals the log of 100 over the percent transmittance. So this will allow you to convert from percent transmittance to absorbance. So this ends our pre-lab lecture for iron analysis. If you have questions, make sure you come to lab and ask them, and you should be good to go.